The first thing you do, more, share screen.
And then we can
Okay. خلاص you're sharing the screen. أيوة. لا, the voice is working خلاص. I think it's good. Now you should speak on the microphone أصلا. What you do more, you're gonna, it's already recording خلاص. Okay.
بعدين إذا خلصت سوي pause.
لا, you don't pause, you end meeting.
Okay.
Okay. خلاص. Okay. That's recording.
Okay. And it's 8 o'clock. Good morning, everybody. Welcome to our histology class. We are doing microscopy and getting ready for you for the lab. Okay, but first I would like to introduce you two wonderful TAs who are going to work with us in the lab. This is Dr. Gadi. And Dr. Malak. And let's get to it. So where's the video? This guy, right? This is the video, right? So let's start with this. Is that?
Zoom. So just, no.
Where? Where do I go?
Yeah.
Oh, okay, it's over there. Okay, so please, you may want to watch this movie later on your own as well. Okay, you see? Turn on the microscope with the power button. Select your slide from the tray, put it on a white sheet of paper, and look at it with your naked eye. Put the slide in onto the stage of the microscope with the stage clicked. After this, you do not need to touch the slide every day. Make sure that you can see some colors so that the tissue is under the slide. So let's do focus. Step number one, we are doing it with the force control. Now, please bring the stage all the way to the bottom as much as you can and clear the knot. You bring it to the maximum. Then after this, you look inside the microscope and start engaging the stage with the force knob and move inside and wait until the images actually becomes clear. And focusing this done. Focusing is done. So all you have to do was elevate the stage until the image was clear. Now you have some structures on a higher magnification. Let's say I would like to look at this dot on a higher magnification. So what do? Please grab the stage control knob and move the slide so that the dot will be in the center, in the middle of the view. Once it's in the center, that's when you are ready to move from the lowest magnification to the next yellow magnification. Now we need to focus. When you are on the yellow magnification, you focus with the fine control knob. And here it is. So it looks like here it is on the top. Now let's say you want to look at this granular tissue on the highest magnification. So what are we doing first? We bring the structure to the middle of the view again with the stage control knob. Do not touch the slide with your hand. So now you are ready to move to the next blue highest magnification blue object. And focus only one and here you can focus image on the presentation form of this plan. So once you are done with the focusing and done with this slide, make sure that you bring the microscope back to the original shape. Meaning you have go back to the shortest objective, remove the slide from the microscope, return it to the tray, and turn off the microscope. So you start microscope on the red objective and finish the microscope. Not to break the slide and the microscope. Thank you.
Okay, guys. I just wanted to tell you the guys that we made just a few days ago this video with Dr. Gadi and Dr. Malak. And actually the words, the subtitles are all made by Dr. Gadi. Okay, so let's get to the microscopy. And let me find where I am quickly. Yeah, here we go. So we're gonna go through all epithelial tissues and gland tissues that I believe Dr. Zafar covered by now. He did, right? Very good. So I gave you a little bit of lecture before in cell biology. What did I tell you we need to focus on most of the time when you look into the microscope? How do you find a cell? How do you identify a cell? Very good. You need to look for the nucleus. So remember, that's exactly what we're gonna do. Scavenger hunt for nuclei all over. Okay, so these are gonna be your 8 slides that you are going to look at in the lab. And you will have, before I start, I also wanted to show you your resources on Moodle. So I believe, I don't know, maybe some of you already looked at it, right? This is the histology section, and under it, histology lab material that is broken down to you, how to use the microscope and the steps of microscopy. That's what we just did. And I also have a printed paper about it, right, in this folder. So one of them is the movie, this is the movie, this is a paper if you like to read. Then the second is gonna be the Olivia guide. And here, there is a Word document here where you're gonna have another 2 videos showing you how to use the virtual microscope, okay? And under this tab, this is the actual step-by-step microscopy printed up on paper form, available for you. So all you have to do in the lab that you just follow the directions and boom, okay? And lab 2 is next week. Okay, so now that we got this straight, let's look at the slides one by one. You guys know when we talk about epithelium in general, we have to talk about simple epithelia, that is only 1 layer. And then we have to talk about stratified epithelia that are made out of many layers of epithelial cells. And what is the third group? This is usually always the hardest one. Glands. Glands are made out of epithelial cells, epithelial tissue. Okay, so we're gonna go down one by one. In the first, we look at you click on this folder. Please do not touch the yellow folder, okay? Do not delete it, don't play with it, nothing, because then your virtual microscope is not gonna work. All you have to do is click on the blue, okay? Let's see how far it is gonna go. And one more word that do you see this little blue icon here? That's the virtual microscope. That's what you need to download on your laptop. Okay, so you can study these slides at home. So what we're gonna do is in the lab, you're gonna use the real microscope. And when you go home, you can repeat with the virtual microscope. Okay, and before I actually get into it, I also wanted to show you, you guys gonna get this. You see here is Olivia 2.9. That is your program that you need to put on your laptop in order to use the microscope. Okay, so here we go. And here's the kidney. Okay, let's see, where are the annotations, meaning all the labels that I created for you guys is here. I'm gonna just try to make it bigger so you see better. It's all in here. You're gonna see two kinds of labels here. You see these little TVs, blue TVs? Those are different views that I saved for you because that's the same structures are all over on the slide. I just thought I'd try to choose for you the best one. So when you click on the blue TV, basically the slide is gonna jump there where I want it to jump. So what you are looking at right now, the whole view of the kidney, right? And then we're gonna look at the three simple epithelium. Guess what? They are all inside kidney. The simple squamous epithelium, the simple cuboidal epithelium, and the simple columnar epithelium. They are all here. So again, let's click on the TV to look at the simple squamous epithelium. And here we go. And you can go full screen. When you keep this here, then you see the labels, including all the words I put here for you. Let's say, what did I label here? And let me show you the full screen. You can go full screen. Looks a little slow, but hopefully it's gonna work faster. Then you put your little mouse there and it tells you what I labeled for you. So what am I, and every time you want to get closer to the structure, all you have to do is roll the mouse. So I'm zooming in so you can see better the flat. So what is the identification of the simple squamous epithelium or squamous epithelial cells? Flat nucleus. And here they are, 1, 2, 3, 4, 5, 6. And in one line. If you don't see nucleus like here, you can see a little bit of cytoplasm, but basically when you are cutting the slide, the knife missed the nucleus. The knife went through the cytoplasm, not the nucleus. You're gonna see this all the time, okay? So basically, this is your flat nucleus. And what is here? Lumen filled with P. So what is this epithelium is actually doing? Holding P. And make sure that the P cannot leak out to the deeper tissues in the kidney. Okay, so let's look at let's go back to the view so that we can look at now, and you can actually do an interactive kind of self-checking. You can close down the labels and open up the labels and you can check yourself, but you still remember what these red flags are. And I hope you saw that anytime let me see. No, that's not what I wanted. That's what I wanted. When I click on the lumen here on the annotation side, then it becomes yellow. So whatever I highlighted here on the image, it's yellow. So you know what is what. Okay, now that we are got this straight, let's look to the simple cuboidal epithelium. And technically we can bring this lower actually. Let me just check. So it's not like, yeah, very good. Simple cuboidal epithelium. Here we go. I jumped in here. And again, anytime you can just roll the mouse and see where you are in the slide. Look, I'm rolling out. This is where I am in the slide. the outside edge of the cortex that we call the cortex. Yes. Okay. So again, let's go back. And if it's cuboid epithelium, what kind of nucleus am I looking for? Very good. In one layer, because simple means one layer. Cuboid means round nucleus. And epithelium means continuous tissue, non-stop at a lumen. So therefore, always find the lumen. Sometimes tubules in the microscope are pushed and you cannot see the lumen. You have to ignore them and move on and find one, okay? So here we go. This is, for example, the lumen of this little tubule, and who is sitting there? P is sitting there again. Because why? Because we are in the kidney, right? And I labeled here for you one round nucleus of the cuboid epithelial cell. But you can see very nicely, this is your epithelium all around the tubule holding the urine. So one thing I want to make sure that you fully understand, everything is typed up like that. The name of the cell, name of the tissue. Please do not mix up the two with each other, okay? So when I ask you in an OSPI question, give me the name of a cell, what are the name of the cells in the epithelium? Squamous epithelial cell, cuboidal epithelial cell, columnar epithelial cell. If I ask you what is the name of the tissue, these are the name of the tissues right here. I even put it there for you. So you see, simple squamous epithelium tissue, blah, 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 blah. Okay? Make sure that you know, remember that. Now let's jump to the simple columnar epithelium. And here we go again. You can see we are at the papilla actually, which is, you know, this little bumpy at the kidney. So we are not in the cortex anymore. You see, the cortex had the simple squamous and the simple cuboid. Okay, so let's jump back there. And what kind of nucleus am I looking for? Oval. But I want to give you a warning. Oval nucleus can, because it's huge, big oval structure, it can appear in two different ways in the microscope. Either if the knife goes through beautifully parallel with the nucleus, longitudinally, then the nucleus is going to look like a oval structure. But if the knife happened to go through this long structure transverse section, what are you going to see? A round structure. So as if it was cuboidal, but it's not. How do I know that it's not? When you see mixture of columnar oval nucleus and mixture of oval nucleus and round nucleus, it cannot be cuboidal. Number one. Number two, I labeled for you. Look, let's go one by one. Lumen P again. Then we go out. This little fuzzy something is the microvilli. We covered it in the lecture. This is the way you're going to see microvilli. Just pink something, pink line, fuzzy something. Then look, this is your columnar epithelial cell right in here with the oval nucleus. And look, the shape of the cell. Is that cuboid or columnar? Huge cytoplasm on the top of the nucleus. The nucleus is kind of pushed to the basement membrane, which is supposed to be here, and huge cytoplasm. So even the size of the cytoplasm is going to tell you that this is it's a columnar cell. Okay, but I also labeled here for you a round nucleus, that where the knife happened to go across. Okay, so we are done with this slide. Let's move to the next one, and we're going to look at the next or the last simple epithelium. You guys remember? That's the mouthful, pseudostratified, columnar, ciliated, epithelium, tissue with goblet cells. Drives you crazy, but actually once you said that, you know everything what there is. So here we go. What you are looking at is the trachea slide. And I labeled for you two things, epithelium, cilia, goblet cell. Here we go. So this is like, you can see that I zoomed into the, let's see where we are. You see, I zoom, this is the lumen, and it has to face an empty space, a lumen, right in here, okay? And this is your epithelium. Now I'm gonna zoom in even more, and here are the labels for you, okay? So this is the lumen in the trachea. What's in the trachea? P, right? No, air, right? Okay, so here's the air breathing in and out. Then the next structure I labeled for you was here is a goblet cell. It looks like a goblet. What is a goblet? A glass that the king holds in his hand. Okay, what else did I label for you? These are gonna be your, I wanted you to remember that mucus covers the cilia. So I put here for you mucus covering cilia, not visible. Just a reminder for you. Then this brushy thing is the cilia. You can see it very nicely. You see how different a microvilli looks like and the cilia, right? Microvilli was just something fuzzy mush. And here is basal bodies of the cilia. And finally, I labeled for you, this would be one columnar epithelial cell with, what did I label here? The cytoplasma. And here I labeled for you the oval nucleus. And you guys remember, I don't want to give lecture about pseudocytes. You guys know that it appears to be multiple layers. That's why it's called pseudo stratified, because you see like 2 or 3 layers of nuclei on top of each other, which is the specialty of stratified epithelium. When you talk about simple epithelium, the nuclei has to be all in one line. And here it isn't. So how did they decide that this is still a simple epithelium? You can't really tell from the light microscope image, but every single epithelial cell that are sitting here on that appears to be on top of each other are all sitting on the same basement membrane. That's why it makes it simple epithelium. Okay, so let's look at the next slide. Okay, let's go back. So remember, no touchy of the yellow folder, no touchy. I also wanted to point out to you, do you see that there is a little horizontal line at the front and horizontal in the back? That actually tells you that this is not any kind of folder. You know, regular yellow folders usually don't have these lines. So that's really tells you that this is part of the virtual microscope. Technically, the programming behind what we are doing is sitting there. So if you get rid of that, touch it, play with it, you're not gonna see a thing. So ignore the yellow line folder with the 2 little mustache. Let's call it mustache. Okay, so let's do esophagus. So what does it mean? We are done with the 4 simple epithelia. Okay. So why do I need thick epithelium in my esophagus? And I guess because we are swallowing, right? And sometimes the food is sharp, hard, and we want to make sure that it's not going to scratch the epithelium. If it was a simple squamous epithelium, we would be bleeding every time we eat something. Let's put it that way, okay? So we need the thick epithelium for swallowing. Okay, so this is the way entire esophagus looks like in, you know, from zoom out. And so this is what we call the esophagus in transverse section, okay? And then you see I labeled 3 different things for you. The first one is, of course, the epithelium, stratified squamous epithelium without keratin. There is no keratin here. Keratin protein fibers are missing. Can they show up here? Yes, they can. If the friction is too strong on the surface of the epithelium, keratin actually can develop for protection. Okay, so this is, so you have to get used to this view, guys, okay? This whole thing is the epithelium, okay? This whole thing, okay? And then comes the connective tissue. So how many layers? How do I know how many layers? What do I have to count? We have to count the nuclei. So we usually start with the bottom, right? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, something like that. Okay. So let's go again, what we labeled here. That this is going to be the lumen where the food is going during swallowing. A reminder again, no keratin, but there is mucus sitting here to lubricate the food. Again, you cannot see it, but I put that for you as a reminder. And here comes the flat nucleus of the squamous epithelial cell that has no keratin on the surface. And guess what? I put here for you, I believe, where is it? No keratin, flat nucleus. Let's just, let me see, where is it? I guess I didn't put it here, but so why is it, let's finish the whole thing. Sorry for interruption. Okay, then comes this one, round nucleus of the cuboidal epithelial cell in the middle of the epithelium. So you see, you have flat nucleus. You have flat nucleus on the top. You have round nucleus in the middle of the tissue. And I labeled here one for you, one oval nucleus on the basement membrane. So where does the terminology of a stratified epithelium is coming from? From the top layer. Because technically the epithelium is a mixture of all three kinds of epithelial cells. The squamous one, the cuboidal one, and the columnar. But the terminology always goes by the top. So if it's squamous, then it's going to be a stratified squamous epithelium. Okay. And what else did I label for you? Because it was so, I couldn't control myself, let's put it that way. You can see very nicely, and we did that in cell biology again. You can see, you see the pink all around is the cytoplasm. Then this purple is the nucleus. And this little cute fellow is the nucleolus. And look, there are darker purple and lighter areas in the nucleus. You guys remember we saw that in the electron microscope. If the lighter one, that's the, very good, the euchromatin, which is active doing transcription. And then when you see the darker condensed DNA, that's the heterochromatin. You see, sometimes you can see it in the light microscope. It depends on how good the slide is. Okay, and I think that's about it. So I labeled for you a nucleolus so that you would know. And here I labeled the cytoplasm for you. Okay, and again, you can look where we are. Let's zoom out. This is where I labeled it for you. But you see the thick epithelium is all around in this kind of star-shaped lumen. Okay, so let's look at now we're gonna look at how a duct looks like, okay? Which is kind of belongs to already to the glands. Glands always have a duct where the secretion is going. But this slide is very good at looking at longitudinal and transverse section of ducts. Okay, so click on the little TV again. And here is a beautiful, let's go full screen. Sometimes I forget. But anyway, you can go back and forth anytime you want. And now you can see very nicely this little whitish, pinkish. Actually, you have different color than I have here. But anyway, yes. So you can see these little flowers. That's really the acini of the glands. What is acini? The secretory part. Those are those epithelial cells. Reminder, glands are always epithelium. Okay, make sure epithelial cells that are actually doing secretion. How does secretion usually occur? We did that in cell biology. Exocytosis. So they're going to exocytosis into this duct. So each one of them has, so here we go, I labeled esophageal gland ocinus looks like a little flower. Then I labeled for you, again, the lumen of the duct. And yes, mucus is sitting here because that's what's going to lubricate the food for you. And what else did I label for you? Yeah, here we go. Let's zoom in here. Yeah. You see, it's so long because I wanted you to see the connection between the ocini of the gland, and the duct is going through the connective tissue and heading toward the epithelium, the stratified epithelium. Then here the duct stops on the image. Does the duct stop here, guys? Where do I need the secretion? Here. So the duct doesn't stop here, guys. Just the knife missed the rest of the duct. This duct is gonna go through the stratified epithelium and is gonna bring the secretion straight to the lumen where the food needs to be lubricated. Okay, so so that's why it's hard to see everything at one shot because the duct is really long. So then here I labeled for you the 2 layers of round nuclei. That what of cuboidal epithelial cells. This is a type of stratified cuboidal epithelium. I like to call it double cuboid to separate from other stratified cuboidal epithelium that has more than 2 layers of round nuclei. Okay, so very good. So that was the duct. How was the duct cut? Longitudinally. Let me see, did I label? Yeah, I also labeled here for you. Now, finally, hello, right? You're gonna be able to see, we talked about the other day, you remember vitamin C, collagen, my university, Nobel Prize, you guys remember that? Now I said, okay, why not, even though this lecture is about epithelium, I said, why don't I label for you, this is the way collagen fibers look like in all kinds of cuts. And who is this fellow? This is just on the side, we didn't cover yet. This is the fibroblast who actually, a cell, living cell, who actually makes the collagen. So why am I bringing this up here? Because you guys remember, I wanted to compare for you cytoskeleton, fibrous proteins, and collagen, fibrous proteins. Where is the cytoskeleton, guys? Inside the cell. So if this is one fibroblast, the cytoskeletal proteins are inside, intracellular proteins. When you look at the collagen, can this guy fit in there? No, it is an extracellular huge protein. That's what I wanted you to grasp, okay? Technically, in classification, both of them are fibrous proteins, okay? But one of them is intracellular, they are tiny inside cell, and this is actually the nucleus, so it's the cell is somewhere here, it's just hard to see. And then the collagen is huge, very strong protein fibers supporting you. And again, I told you, this protein is everywhere. That's why you need vitamin C, because if vitamin breaks down, if you don't have enough, then collagen breaks down, and collagen is everywhere. So technically every organ will shut down, with a little exaggeration, but it's really true. Okay, finally, let's look at the same thing. Just we're gonna look at the cross section of a duct. Here we go. Okay, I labeled for you. You see here's one little flower, ocinus. I call these ocini when they are cut across little flowers, you see? So these are the epithelial cells that secrete into this little duct in the center by exocytosis. Here's another one. You can see the duct a little bit better here. But my purpose here was not to label glands for you. So that's why, let's just go back here. So what do I look at now? A cross section or transverse section of a duct. The same thing what you saw longitudinal. So this is the lumen of the duct, okay? And look at the 2 layers of I labeled it here for you, 2 layers of round nuclei, cuboidal epithelial cells. Okay, so let's move on. And what is the next? Esophagus is done. Okay, now we're gonna look at thin skin. But if I say an OSPI, what is the name of the tissue in thin skin? You don't see thin skin, okay? What are you gonna say? I'm gonna show it to you. Dr. Zafar covered it, I'm sure. It's gonna be stratified, squamous, epithelium with thin keratin on the top. Still opening up. Here we go. So this is the way you see it from far away. Let's close this flex down. And here we go. This is your thin skin. This is the way it looks like when I get a little bit closer to the surface. And I want to make sure again that you understand this entire purple thing is the epithelium. And this very thin little fibers falling off at the surface is the thin keratin. Okay, so now I show you what we labeled here. It's basically identical with what we just did in the esophagus, just with thin keratin. Okay, so here we go. Let me jump where I labeled it for you. We can go full screen. And I went from outside. This is your outside world. It's not lumen anymore. This is this. Okay, because we are doing with skin. Okay, then this is your thin keratin falling off or desquamation. You guys know it's keep falling all day long. This is the dust in the house, dandruff, in the rest of the world. In Saudi Arabia, it's dust from the Sahara. But here, everywhere else, the dust is from keratin. Okay, now what did I label here? A flat nucleus of the squamous epithelial cell. And now here, I meant to do in the previous slide, that's where the terminology is coming from. You remember, from the top layer. Then you go a little bit deeper. I labeled here for you the round nucleus in the middle of the epithelium. I labeled here for you the oval nucleus on the basement membrane. And again, I said I can't pass to label for you this little cute nucleolus in the nucleus. You see on this slide, you don't really see very well the hetero and the euchromatin. Maybe here, maybe a little bit here, but not as clear in the other one. And finally, cytoplasma. And you remember, plasma membrane is usually you don't see. Basement membrane, usually you don't see. That's why we're really focusing on the, I mean, what am I saying? Yes, you can see the cytoplasma as pink, of course, but you're not gonna see most of the time, the borders of the cells, okay? So that was the skin thing. And let's move on to the thick skin. And thin skin is technically all over our body. The thick skin is the one that is only in 2 locations, on the hands and the feet. Why? Because of friction. So that's why you have most keratin here. I put here for you 2 different kind of slides. Two versions, and both of them are going to be in the laboratory, but we have more of this slide than the second one. So here we go. Plantar skin, very good. So thick skin, and again, that's not, this is the way it looks like. Okay, when I zoom in, and again, remember, these are all the epithelial cells. This whole thing is the epithelium. Again, if I want to know how many layers, what do? Start counting the nuclei. Boom, 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 right? And this is the really, really, really thick keratin. Keratin is thicker than some parts of the epithelial cells layers, all together. Okay, now that we got this straight, let's go to the labels. So what is the epithelium? What is the name of this epithelium? Stratified squamous epithelium with thick keratin tissue. Okay, so again, I put that tissue for you, so make sure we don't mix the tissue names and the cell names with each other. Okay, so let's jump there. And this is what I labeled for you. Okay, so this is the outside world again. This is the thick keratin. Okay, wait a minute, we can go full screen again. Looks nicer. Then this is the flat nucleus. You can see many of them of the squamous epithelial cell on the top, where the terminology is coming from. You remember epithelium, epi means, what does it mean epi? On the top. So you always have to find it at some free space, outside world or some kind of cavity lumen inside body. Okay, then I labeled here for you the, okay, let's move a little bit closer. I labeled here for you the round nucleus of a cuboid looking epithelial cell in the middle. And this is what I labeled for you, the oval nucleus of the columnar epithelial cells that are sitting on the basement membrane here. Okay, so that's what the basement membrane is. And observe that some images, the free space is on the bottom, some images on the top. And I prefer to put the lumen and the free space on the top, but that's one limitation of this microscope. The way they set it up, the scanning and digitalizing, I cannot do that. It's just between you and me. But let's say I want to make an exam question about this image. I would probably put the keratin on the top. You understand? So do not get lost that the epithelium is showing you on the bottom or sometimes on the top. Okay. What else did I label for you? Again, collagen fibers. Collagen fibers. So who are these cells all around? Many of them fibroblasts, right? And again, I labeled for you nucleus. This is the nucleolus in the nucleus, and I labeled for you cytoplasm. So you see, it's really the same thing, what we just did. Okay, so let's go to the second version of the plantar skin. And I wanted to show you something interesting about this slide. Every slide has some, you know, benefits or curses. Looks good, doesn't look good. What am I gonna do? You guys just have to get used to it. The quality of the slide can vary. Even inside the same slide, some area on the left is gonna be blurry because it's blurry. The slide is probably too thick. On the other side, it's beautiful, clear. So what are you gonna do? Move to the clear side and ignore the blurry, okay? So let's see, this is the thick skin. Let's go there. Look at this. Is it thick enough for you? This is really the epithelial cells in many layers. And this big pink thing is the thick keratin. This whole pink thing. Okay, so what is the name again of this tissue? Stratified squamous epithelium with thick keratin tissue, and I zoomed in. Okay, so let's go one by one. Outside world, thick keratin, and this is the stratified squamous epithelium, okay? And then let's go there. And what did I label? You see, now you cannot see the outside world because the keratin is too thick. So when you zoom in, the outside world will go away. But let's go one by one. This is the thick keratin, flat nucleus on the top again. And round nucleus in the middle. Oval nucleus in the base, on the basement membrane. I labeled for you again collagen and collagen. I also labeled for you nucleolus in the nucleus, but there's two more structures. Do you guys see this little, looks like little zebra between the cells, these lines? Do you see this? Okay, thank you for answering my question. The maximum you can go to when you go to view Maximum you can go to 40. So I am on 40, so I cannot make it bigger than this. So what are those little zebra looking lines around the cells? Desmosomes. Hello, cell biology. We did the desmosomes. This is the way it looks in light microscope. Okay, so let's move on to the next slide. What is missing so far is the, let me see. So we did the two plantar skin, rabbit of the penis, penis of the rabbit. It's very important that you orient yourself because the entire structure is there. You see? This whole thing. So you have to orient yourself here. But what is really interesting that this is a thin skin stratified squamous, thin keratin tissue here around the surface. Guess what? There is a piece of bone in the middle. Funny. And then this is basically, what am I doing? This little long lumen is the urethra. So this is where the stratified columnar epithelium tissue sits. Okay? So you click here and it's gonna take you right here, labeling the lumen where the pea or sperm can sit. This is a round nucleus. This is an oval nucleus because you remember columnar always can alternate. Then again, oval and oval. How many layers do we have here? Right here is 1, 2, and 3. If you go here, maybe 4 or 5. And collagen fibers. At night, it's already boring, right? We did it so many times. Okay, let's look at the ureter. So this is where you're gonna find the true stratified cuboidal epithelium. So what are you anticipating? At the surface, at the lumen, the nucleus is, if it's cuboidal. Okay, so here it is, the whole view. It almost looks like esophagus, but it isn't. Okay, because there are no glands, just observe, no glands and ducts. Okay, what I showed you. So let's look at the actual epithelium. So this is the epithelium. Observe the lumen where the P is. This is the round nucleus of the cuboidal epithelial cell at the surface. They are huge, so some people call them dome shape. Then here's more round nucleus, oval nucleus, collagen. Now wait a minute, here I labeled for you the boom, boom, boom. This is the boom 1 is the fibroblast. Boom 2 is the collagen. Boom 3, ground substance. The white nothing, which is basically interstitial fluid, extracellular fluid. Okay, and finally, let's look at the glands. I like to teach glands, guys, on duodenum slide because you're gonna see a lot of glands. You already actually saw one in the esophagus, right? But on this one, you can see 3 kinds on top of each other. So I think it's a blessing. So we don't have to continuously, so it's to begin to learn about glands, I think it's a perfect slide. Wake up. So what are the glands we are looking at here? Goblet cell, which is a unicellular gland. Then simple glands and compound glands. Did Dr. Zafar cover? Okay, then here we go. So there is a unicellular gland, questions later, unicellular gland, which is basically one cell. That's the goblet cell. Then just watch what you are saying, simple tubular gland. It means one little tubule. That's what it means. Compound tubular, many tubules. Okay, branching to make a bigger gland. Okay, ready? So we are in the duodenum. So if you want to orient yourself, here's the, let's go full screen. Again, the tissue is upside down. So this is the lumen where the digestion is going on. I didn't want to put that poop, but that's what it is over there, okay? So look at you can already see very nicely on this slide, the layers, okay? So these layers here, do you see the little fingers there? Those are all the layers of the villi. This is where I labeled for you goblet cells. Now look at under the villi, which is technically this image is above the villi. This is where we can count how many tubules are sitting in here. Let's say 100. This is where the layer of the simple tubular glands are, okay? And finally, right under or above, depends on how the slide is, these circles are all compound glands, okay? So this is one compound gland. So it's branched to little pieces. And look, what is that? Even a duct is visible. Okay, so let's look at quickly what I labeled for you. And then we are done for today. Okay, so let's say gland, no, wait a minute, that's not what I wanted. I wanted gland 1. You see, I labeled for you goblet cell on the top of the villi. And then the tubular glands, which is gland 2, you can see, and the compound glands, gland 3. But it's basically you are going deeper and deeper into the tissue from the lumen. Okay, so gland 1, here we go. This is your goblet. First of all, this is your lumen. This is your goblet cell. You see this pink line? That's microvilli over nucleus of the columnar epithelial cell. So what do you really see? The goblet cell is a lonely little fellow alone, sitting in the middle of the simple columnar epithelium. And I give you a secret, goblet cell is always in columnar epithelium, simple columnar epithelium, which is basically here or the pseudostratified, okay? This is broken tissue, ignore, connective tissue. Okay, let's look at gland number 2. Here we go. Look at how many tubes do you see right now? Tubules. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Let's say 20 tubes. However, observe that some of the lumen is not open. We need the lumen. Okay, so ignore those tubules. Like let's say this is a tubule. It's closed. It got shut down during processing. So you have to choose one that is actually open, because otherwise you don't know what's going on. So here we go. So I selected this one tube for you. This is another gland, okay? This is another gland. This is another gland. So individual little tubules alone, parallel, sitting next to each other. No touchy, okay? So here is lumen. Microvilli, again, this very thin pink line. Over nucleus of the columnar epithelial cell. So what are we looking at again? Simple columnar epithelial. So who makes the gland, guys? Simple columnar epithelial tissue makes the gland. This is it. The whole thing, okay? And when you are at the base of the duct, of the gland, this is what you call acinos. These guys, these epithelial cells actually secrete how? Exocytosis. And look, there is even goblet cell in here. Then now the secretion is gonna go into the lumen by exocytosis, traveling, until you hit the duct. So that the top part of the gland is considered to be the duct. And look, even goblet cell appears here. This is what I call gland in the gland. What does it mean? Goblet cell gland is in the simple tubular gland. That's what it means. And the secretion is gonna go, you can't always see it, between the, you can see actually here, between the villi, the secretion is gonna go into the lumen of the duodenum. And finally, let's look at the compound gland. And it looks like my time is up, so let's hurry up. Yeah, I labeled for you one gland, by the way, here in cross section. If you cut the tube across, you're gonna see a circle, right? Here's your lumen, here's the microvilli, and here are the columnar epithelial cells. Okay, finally, these are the compound tubular glands. How many? 1, 2, 3, 4, 5 right now. And I label, what did I label for you? These are your acini. This is the epithelium, simple columnar epithelium with microvilli. This is the lumen of the duct. And this is the, again, simple columnar epithelium with microvilli in the duct. Okay, and the duct is gonna again bring the secretion all the way between the villi to the lumen of the duodenum. The end. Okay. All right. Okay, I just would like to get 3 volunteers from F1, F2, and male, who is supposed to be the leader of the groups. Come and talk to me or come to my office or email me. I want to let you know how you guys gonna get access to this material, okay? What about what? Oh my God. Yes, attendance. Are you guys ready? Attendance. So today is September 6th. Wake up. It's very slow. Okay, tell me which one is us. This one, 8 a.m., right? And all students, yes, yes. But there's no arrow here. Okay, here we go. Very slow. So where's the thing generate? Where is the generate? Can you guys see it? Where is it? Still loading. And then whoever turned on the Zoom, come up there and shut it off, please, because Dr. Junaid is here. Okay, I guess we're gonna do attendance next time, okay? Remind me. Where is it? It's very slow. Where? Where? That's what I was. Exactly, that's what I was looking for. But I don't, what does that do?
Basically, it makes everyone who has his own internet can scan. But yes, makes it only the people connected to the faceless internet.
Okay, guys. I have to also exit this thing. So I want everything to shut down. Everything shut down. Yeah. Remove. Remove. And the virtual microscope, everything. But that's just
when the
Finishes.
yeah, when the was everything okay with the IT?
yeah, everything was fine. Yes.
Can we generate another code without the restriction of the connection?
No, because Jonah is here. We do it next time. Or I just do it in the office. Everybody was
We
here.
asked the PLCs, we will send
Okay,
it
okay,
email.
I need to exit. I need to exit, guys.
Yes.
What is this?
This is the
this is the
meeting.
Zoom.
Also.
Okay.
You want to
Okay.
remove the
I
USB?
wanna, yes, could you eject
yes,
the
yes.
USB? This one you shut down. And then,
Here. خلاص بعدين
We will make a form again. We will send it to the doctor.
I ejected.
how do I know it's, no, it's not. It is ejected. Is it ejected? It doesn't show, right?
Yes.
Okay.
Okay. Thank you. تعال.
This didn't connect to Wi-Fi, so it expired.
You can send on the Google Form. On the meeting, you can do course.
I apologize.
ما في سكرين شوت ما اخذنا